the cellular localizations of human endogenous retrovirus in colorectal cancer cells. The outline for my presentation for today is I will talk about why am I focusing on colorectal cancer and what is human endogenous retrovirus all about. And of course, I will share with you later my experimental approaches and the results. Of course, at the end of my presentation, I would like to acknowledge those who have helped me in this particular project. So, colorectal cancer. Why am I focusing on colorectal cancer? Because colorectal cancer is the third most commonly occurring cancer worldwide. And based on this data from WHO, it's Global Can 2008. Believe me, this is the latest. And this is we can see that 10%, okay, which is over here, it ranks as the third in the incidence rates for cancer. Back in Singapore, you can see that it has top itself, okay, has been the 18% in terms of the incidence rates. That is actually for 2008. If you are looking for a period of time, over here, we are looking, let's say we look at from 2007 to 2011, we can see that for the males, they are still top as being the commonest cancer, and for the females, it ranks second, right after female breast cancer. So as you can see from here is that we are treating colorectal cancer a concern worldwide and also in the local aspect. So that is why we are wanted to find out is there any novel player in colorectal carcinogenesis. So that's why we have take a look at human endogenous retrovirus. Before I talk about what is human endogenous retrovirus, perhaps you shall recap on what is retrovirus. Now, retrovirus, as we all would have probably known, that it is an RNA virus. One of the famous or infamous example would be HIV. And what it do is that it will infect the human cells, and what does it do is that it not only it can infect, it can also incorporate its proviral DNA into the genome of the cells. And of course, upon appropriate stimulation and with the host machinery, it is able to generate a new generation of virus. So this is the aspect of exogenous retrovirus. So from here, what is endogenous retrovirus? Well, that would mean that is the footprint or the remains of this ancient retroviral infection of the human genome. And this infection has occurred at least 30 million years ago. So from here, what does that mean? Is that at least 30 million years ago, this exogenous retrovirus is that it has overcome the retrovirus restriction factors, for example, shrimp 5-alpha, and it has successfully infected the germline cells and incorporated its proviral DNA into the genome. And as a result of this endogenation process, right now the proviral is able to transmit vertically, such as any other genes. Such as any other genes, okay, and it will be transmitted like a medallion manners. So I hope that I'm not giving you a sci-fi story or records of the X-Files, but this is actually reported in 2001, whereby there is the first report of the Human Genome Project. It is actually reported that 8% of the human genome contains retrovirus-like elements. And over here, what we are seeing that this arrow here is the LTR, which is refers to long terminal repeats, and of course we can have the complete or near complete versions or the truncated versions. So what does this retrovirus, the general structure will be like is this. Of course, it's very similar to a retrovirus, and over here we have the GAG, Pro, Po, and EMB genes. Of course, GAG, of course, it refers to the GAG encodes for all these matrix capsid proteins, and of course, Po is meaning the protease. Po means all the polymerase is necessary for viral integration and replication, and of course, for EMB, referring to those envelopes proteins. And of course, as you can see from here, they are framed on both sides by LTR, which refers to as the long terminal repeats. And this long terminal repeats contains motif that is necessary for the regulation of retroviral gene expressions. So right now, having talked about Herv, which means human endogenous retrovirus, let us move on to the classifications. Is there a standard classification? No. According to ICTF, there is no classification. What is ICTF? It actually refers to as the International Committee on the Taxonomy of Viruses. So in the field, there is two classifications. One of the classification is based on the genetic similarity in the regions, and that can be classified into class 1, class 2, and class 3. So of course, it can be gamma retrovirus like, beta retrovirus like, or spuma retrovirus like. So over here, my focus will be on Herv-H, and so therefore it is actually on the class 1. On the other hand, there is also another more common way of classifying the Herv, okay, which is based on its tRNA specificity. What does that mean is that the H here, which means is that the Herv will use histidine tRNA at its primer binding site for the viral integrations. So, as I mentioned, is that I'm actually much focusing on Herv-H, which is one of the Herv families, and Herv-H itself is abundant. It's widespread in our human genome, and it actually occurs in chromosome 1, 2, 3, 6, 7, 9, 11, 14, 19, and X. And together, they have 1,306 copy numbers in the human genome. And of course, I'm actually much interested in the X chromosomes. The one that is found in X chromosomes, and therefore it still has the general structure of this GAG, Po, EMB that are framed by the LTR, which is bringing the long terminal repeats. So you may wonder, why am I so concerned about Herv-H? That's actually because of some previous study that has been done. It has been shown that Herv-H is selectively expressed in colorectal cancer, but not in normal tissues. So what are those studies? In 2004, using suppressions, subtractive hybridization, they realized one thing is that there is an increased transcript level of Herv-H in adenoma and tumor centers, but not in corresponding normal tissues. Some years later, that is in 2008, it was also found that through this large-scale analysis of gene expressions, they realized is that the Herv-H family is selectively expressed in colorectal cancer but not in normal tissue. And 1 year later, a China group have also found out that this Herv-H is expressed in colorectal tumor tissues but not in the normal tissues. So with that, we are very excited over this. Could this be a new novel players in this colorectal carcinogenesis? So we are trying to find out more. So we have to use an in vitro model, and so therefore we have to look at the cell line. And over here we have found out that for HT29 cell line, which is a colorectal adenocarcinoma cell line, it does not express Herv-H. So therefore, it could be a good candidate for us, okay, to study what is the effect of expression of Herv-H. So of course, HD29 is definitely available from ATCC, which is the American Type Culture Collections. Of course, due to resource limitations, we actually approached the tissue repository at NUHS for a sample of it. So of course, I'm actually grateful to Dr. Ng for provision of this HD29 cell line. So from there, we are aware of these Herv-H expressions based on chromosome XP22, and over of this whole range of the transcripts, we actually realize is that there could be a putative protein that is of 93 amino acids. And over here, that is more like a gap proteins. So with this gap protein that could be synthesized by the colorectal cancer, be involved in any of the mechanism that is involved. Could it be involved in the inflammations, and so therefore there is this kind of inflammation associated colorectal carcinogenesis, or could it be it itself is a kind of like a cancer player whereby it actually binds to the cancer genes, or if not, it can act as a super antigen and thereby creating the inflammation process. So having said all this, is that we are also mindful that the increased expression of the Herv-H elements may or may not be the causative agent, because it could be the consequence. But nevertheless, what we have gone through is that we want to find out, first of all, where is this Herv-H, if it is actually being expressed in the cells. So therefore, we would like to look at the subcellular localizations in the colorectal cells. So what we have done is that we have made good use of the expression factor, which means the plasmids, and specifically we are using these PEGFP-N2. The EGFP refers to enhanced green fluorescent proteins, and this will give us a very beautiful green picture. And over here, as we can see from here, there is this MCS sections. This is the multiple cloning site. So what we are doing is that we insert our Herv-H, that's of these 93 amino acids, into this region here, and that is tagged with EGFP. So we will exactly know where does it localize in the cells. So over here, we have successfully cloned it in, and this is the one whole open reading frame, and thereby we are ready to start to put in this expression factor into the cells. So what we use is that we use this lipofactorins, which is a transfection agent, lipofactorin 2000 CT. By the way, I don't got any financial support from them, so let's move on. Alright, so this is HD29 cells. So over here, what we are seeing is that on the left-hand side, this is the normal cells, and it has not been transfected. So using Herv 33342, which is the DNA dye, it will actually stain out the DNA, and as a result, it will give us the demarcations of the nuclear regions. So before we do on using the Herv-H GFP factor, we would have to transfect it with the empty plasmids. So over here, what we are saying is that this HD29 cell has been transfected with the empty plasmids, which means only the EGFP. And what we can see over here is that we can still see the large nucleus, and over here will be this GFP, and we can see that the GFP is widespread. And looking at more picture, what we are seeing here is that you can see that this is the first staining, which show the nuclear staining, and from there this is just purely GFP, and with a merged image, we can see that our HD29 cells is able to take in the expression factor, and thereby we are then able to move on to see where is our Herv-H localized within the cells. So this will be the ones. So over here, this is still the Herv, first staining, and over here this is the Herv-H that is tagged with GFP. And what we are seeing here is that it is actually on the outside of the nucleus. And we merge it, you can see that it is in the cytoplasm. So moving on, okay, with a better pictures over here, you can see that the HTT9 cells, okay, this is the nucleus staining again, and this is the Herv-H tagged with a GFP. And you can see that, okay, Herv-H is actually localized in the cytoplasm. So with a merged one, with a merged image, okay, it shows even clearly. So over here, what we have shown here is that Herv-H does localize in cytoplasm in stably expressing cell lines. So in our future approach is that we like to find out what does this Herv-H actually do or involve in colorectal cancer carcinogenesis. And we, of course, we have found that Herv-H is actually able to increase the cell proliferation rate, which I have actually reported in my poster presentation outside. So from here, I would like to end my presentations. Okay, with acknowledgements. I would like to thank my supervisor, Prof. Coy and Simon at NUHS, and of course Dr. Eng for providing me the HD25 cell line. And of course, over here at SP, okay, my bosses, Adrian and my wife, for giving me the great support, okay, to do all the work, really to show me the ropes in doing all the tissue culture work initially. Of course, our technical support staff, Dr. Kelvin, and my 2 students. Last but not least, the grant, okay, from Singapore Poly, to make that this project is a success. All right, thanks.